Welcome, everyone. Welcome to the ISNR Webinar Series. Today is June 26, 2026. I'm your moderator, Cynthia Wilson, today. And today we have a sponsored webinar through Vielight, and they have sponsored all of your registrations today, so thank you for that, guys. They will be talking today about precision EEG and guided photobiomodulation, leveraging AI for personalized neuroregulation. We do have three presenters today. We have Dr. Lu Lim, a doctor of natural medicine and a pioneer in photobiomodulation. We have Dr. Riza Moriarty, a neuroscientist and senior scientist at the Brain Stimulation Center, and Dr. Marazi Zaire, computational neuroscience and AI strategist. Before we get started, we do have some housekeeping, so please put all of your questions either in the chat box or in the Q&A. We do have some people that are moderating those questions as we go through this, and then if we have time at the end, we will go through any questions that are left over. We will do our best to get through as many questions as we can. Welcome, Dr. Lim.
Thank you. Well, first of all, we are somewhat overwhelmed by the response to what we're going to share. Thank you, everybody, for joining us. And we will have a recording. So I know that, this recorded. So I just want to say that those who are not able to join here can have a copy of that. And so this webinar is meant to be educational. We're not going to overtly, promote that, although many of you are already familiar with it and with our name. And imagine if you have something that can actually stimulate the brain and make it better. So you have something really useful in your hands, right? Now, it could be electrical, it could be anything. It could be, the nootropics you're taking. But I say that photobiomodulation probably has more evidence than most other form of stimulation. And many of you have seen it. Now, if we partner that with a tool that can collect data from something you're familiar with, like EEG, that gives us something more to work on, with that. And I'll say that, so we are having it. It's going to look interesting, and I'm going to share my screen. I have co-presenters here. Reza is from the University of Toronto. He's really an expert in EEG. I will put him up there at the very top based on totally unbiased opinion. And Mazier has been leading the AI work at Vielight here. Now, I did mention what if you have something that really works on the brain. Now, there are many forms of stimulation that can claim electrical stimulation, particularly, can show you the same thing here. In our case, we use the photon. There is receptor, the chromophore works on it. And the key is always been discussed, mitochondria. I can activate mitochondria in various methods. But I'll say there's more evidence in photobiomodulation than most other methods of activating mitochondria, particularly non-invasively. Then it goes into the signaling that, from activating the various relevant proteins, and then get into the affecting the function of the brain. So this is a simple schematic that I'll say you can go to all the reviews and literature, and you can actually see photobiomodulation probably working better than most other methods. Now, the photobiomodulation is today's red light therapy. It's used everywhere on face masks and panels and everything. Here's where I think everyone has to be discerning here, because you can go up to the sun, you probably get a lot of stimulation, you can get a panel and red light and maybe think that it works. But really, based on research, these different variables are important. You start with a wavelength. What wavelength do you use? It's got to be within the action spectrum, and generally we use 810 nanometers. We are, of course, exploring. We got quite a lot of data on other wavelengths as well. And then you have to know where you want to position the stimulation point. Then the irradiance is like, what is the power that's landed on your skin that affects the outcome. Then duration, how long do you need to do it for? Then we also pulse it to affect EEG, but actually it's to affect the physiological outcomes. Then you get a dose. Now the dose is actually a collection of these various parameters, but it's got to be within a certain window. Too little, particularly the irradiance, the power that lands on the target, doesn't do anything. Too much can have a negative effect. So all these matter, and that's why going into something to help us like machine learning and AI is important. What do we do to give us some indications to guide us? And we thought EEG is really convenient. Many of you are really familiar with that. You also have equipment. It could be a home use EEG equipment, or it can be lab, even in high quality, high resolution EEG. electrodes to measure that. Then we can use this to guide us. Now, there are two paths to the efficacy. Photobiomodulation on its own actually works really well. There are several, should be more than 10,000 pieces of literature out there already. If you can direct it with the right kind of power particularly for the brain, to stimulate the brain as opposed to, say, on your body, on joints, require actually more specialized knowledge or specific application, because you really do need a certain level of power to penetrate. And then the other part, like I mentioned, if we have EEG data, it can guide us based on the EEG profile and actually fine-tune it. So the first one is pretty broad. It generally works. Second one is getting the brain to function at the more precise and at a higher level. Now, there are ways that you can imagine, but I think most of you probably have figured out, if we want to be able to use artificial intelligence, I mentioned the tools, but why do we need artificial intelligence? It allows us to collect the EEG, do the AI analysis in the back end, and then select the parameters, go deliver it. Then you get EEG feedback telling us where the brain state is at that time, and then teaching us what to do next. And then we keep doing it in a loop. I showed here an example of how this can be enabled, and you can experiment many ways, but it's taken us many years and a lot of resources to propose something that's, first of all, convenient, easy to use with a phone, and then to affect the parameters on this device. Now we are entering a new age. I believe it's just beginnings. It's exciting. We have a long way to go. The potential seems limitless, but I want to stress that this is just the beginning. What we're going to show you is just a beginning, and it's kind of available now, but we want to be fully tested. So you'll be invited to be part of this adventure with us. Now, once again, what do we see from the clinical perspective? You measure, you personalize, you track against the norms and apply it in clinical practice. So we are trying to also fill a gap in putting something that clinical practices need to improve the outcomes. So I'm going to hand over to Reza, who will continue the presentation. Thank you.
Thank you, Lou. Thank you, everyone, for attending this webinar. And so the idea of this webinar is sharing a study we are running at Vielight, and to emphasize the importance of personalization of photobiomodulation. And I'm going to relatively give a little long background why we need that and why we need the personalized photobiomodulation. The word is right now in every field. We are saying the personalized and precise intervention or medicine, but we will see we need it even more for photobiomodulation. Photobiomodulation is a new brain stimulation technology that targets the brain, and it has been recently added to the field of the brain stimulation. So before, we only looking at the brain through the electrochemical mechanism. So then that was the language of the brain. And if we wanted to intervene with the brain, so we had two options: through the chemical, drug, pharma, and electro through the electrical or magnetic stimulation. And we didn't have any position for light intervention to the brain. So it has not been very long time ago, like say this is the paper on Nature 2018, they don't have any place for photobiomodulation. But recently, researchers starting to appreciate the presence of the photobiomodulation, and they acknowledge the science of that and importance of photobiomodulation. And right now we have more options: electrical stimulation, magnetic stimulation, pharma, of course, for chemical, sound, and light. This is a new paper, but one of the pioneer in brain stimulation, Andre Brunoni, last year, they put the light in the special category. And one of the important part of the brain stimulation, resolution and the duration, or the temporal or the spatial. So far we know much about the electrical or magnetic stimulation and also sound, ultrasound recently, and also the pharma. It seems the photobiomodulation have a special place here in terms of the range, resolution, in terms of the location, or the extent of the application, and also in time. We will go back over that time in the next slides. A photobiomodulation or light, it's very simple technology compared to electrical, magnetic, or even ultrasound. And I listed some advantage here. It's really easy to set up, so people can purchase it and go it at home. And the training is very minimum. Operational is very easy. Portability, It's a take-home device. You can go to trip with it, and compared to other electrical or brain stimulation, it is much easier to use. But the mechanism still is under investigation, and we have several ideas how the photobiomodulation interacts with brain. It's very important to understand what we are talking here and why the science of photobiomodulation could be more complex than electrical or magnetic or other brain intervention that currently practiced in clinical work. Mitochondria is one of the main resource for photobiomodulation. That's one of the very well accepted theory, acting on the calcium channels, increasing the blood flow, even working on the level of the microtubule. And people are even starting to think photobiomodulation is not only starting the chain of the chemical reaction. Sorry about that. So right now we have different modality that interacts with the brain through the electrical or magnetic and light. We're talking more complex mechanism, but it gives us anyway the option to modulate neural network, enhancing or suppressing the brain network. And for suppressing or enhancing the brain network, it's very important topics in the neurophysiology and the clinical application. First, I will go over the TMS part, which is probably most of you know about that. With the TMS, the mechanism of the TMS is actually generate the action potential. It fires the cell. But how we are delivering that pulse, single pulse over 1 second, 2 seconds, or even 5 seconds, or even we can send it few pulses in different milliseconds, they have different meaning and different interaction with the brain. So based off how we deliver the magnetic field, how we deliver the electrical induced by magnetic field to brain, we can have with the excitatory or inhibitory protocols. One of the famous in the clinical application, low frequency 1 Hertz, call it inhibitory protocol, or excitatory protocols when we are delivering with the high frequency. Usually low frequency, we are going for the anxiety, high frequency, we are going for the depression. And famous protocol of the ITBS, it's also have 2 mode of the inhibitory and excitatory, clearly a very well-known protocol applied for the clinical application. And with the delivering of the pulse, we can create really, this is over the motor cortex, we can create the excitation or inhibition. It is very well studied. And with the TMS, still they are exploring that part, but we know by inducing the electrical stimulation or stimulating one location of the brain, we can enhance or suppress the activity in that. I'm going over only one famous slides here for ITBS that is developed by Professor John Roswell. And they show it by delivering in certain pattern of the electrical magnetic stimulation, we can generate an excitatory or inhibitory effect on the brain. So they call it ITBS or CTBS. That's 2 famous pattern of the delivering the magnetic field. Compared to the baseline, you deliver it for 3 minutes, then over 20 minutes or 30 minutes, you will have the excitatory response and you will have the inhibitory response. This protocol developed by group and John Roswell's lab in 2005. Later, the same group, they trying to reevaluate what's happening if we reproduce this result. They reproduce this result and I see that's the result A and B. It's very hard to guess which protocol is excitatory, which protocol is inhibitory. Here in the previous publication, they have the ITBS as an excitatory, CTBS as an inhibitory, but a few years later, they reproduced the result and they said, no, that's not that easy. And it's really hard to guess which condition is excitatory or inhibitory. And, this is the inhibitory protocol, this is excitatory protocol. And they concluded the variation in the RTMS plasticity, it really depends which interneural network we are recruiting. If we don't know, if it is too variable, we will have very variable results. And it's not easy to reproduce excitatory or inhibitory. Later, scientists, clinicians, they trying to understand this mechanism and coming with the solution. And in Professor Siemens' lab, Christoph Zerner's coming with very nice idea. He said, when we wanted to stimulate the motor cortex and we don't know what is the underlying mechanism, simple idea, very brilliant, sending the TMS pulse, syncing with the alpha or the mu rhythm at the motor cortex. If we're detecting the ongoing oscillation, if it is at the peak, we are sending 1 pulse. If it is at trough, we are sending 1 pulse. So it's a state-dependent brain. And they find it, yeah, this is quite successful. If we can manage it to detect the ongoing alpha oscillation or mu rhythm close to motor cortex and send the pulse at very special time, then we will have much more reliable and reproducible response. So it's like state dependency started and closed loop stimulation started by Christoph Zerner. And they proposed that idea that state dependency is important. Also, the protocol could be excitatory or inhibitory, but it's very dependent on the state of the brain. We have the same finding for TDCS, another famous brain stimulation technology. We have anodal and cathodal stimulation to very weak electrical stimulation. And generally, we believe it has been shown when you stimulated the brain region with the positive current or polarity, you will have the excitation, as you will see it here in figure A. You have the excitation. If you put the negative part or negative current, you will have the inhibition. So another protocol for excitation or inhibition, we hope we could come with the transcranial electrical stimulation. As you may guess, result trying to reproduce again by Dr. Ruswell, and they find it quite variable. It's not easy to understand or to guess which of this protocol is a positive current or which one is negative current. And they had a lot of idea why we have this variability. It's time dependent. When we are recording that, what is the brain state? And this is a really fascinating paper that they talk about the brain variability and response. So also, we have a good idea of excitation or inhibition in brain, but it's still challenging, even with very well-studied tools. So then all of this intervention becomes more complex now. And all the advanced neurophysiology or the brain stimulation clinic research or university, they are focusing on optimizing. Optimizing means let's do it for each person. And if we wanted to come with the excitation or inhibition, it's not like a simple assumption of the low frequency, high frequency. It's not that easy. We have to understand what is a neuroanatomy. We have to specify for each person. And does it also depends to the state? Why we always have to stimulate it during rest? Maybe we engage the participant in task or provoke some symptom, then we stimulate the brain. And they have done a lot of MRI study and still going. And also they add the computational modeling. Said, yeah, the neuroanatomy is important. First, we have to understand if we are stimulating the brain, which part of the brain is really become excited, inhibited. Based off that, we optimize the coil position or electrode position and even the intensity of current and the duration, and still very active field of optimization. And another method that's by clinician, by Sean Sedgwick and Michael Fox, they are from Harvard. They come with very nice idea how to optimize the RTMS treatment for circuit-based neuromodulation. So it's every brain stimulation technology, they are working on optimization because they found out recently it's not easy. We have some idea, we have some theoretical idea how this stimulation is working, but brain itself is more complex. We don't know that how to control that variability and how to use the technology to modify and to push the brain to direction we want. And of course, people didn't stay there. They are using more fancy and more advanced technology. They are saying, okay, let's use it tractography on real time. When you are putting the coil or stimulation on the real time, you want to see how they are connected with the different area. Maybe that helps us to reach deeper and to other side of the brain and to controlling the optimization for the treatment. And again, more technology is coming that maybe one coil is not enough. Maybe we need the robot to make it more precise stimulation. Maybe we have to target the brain from different location. It's not one location. It's not enough for one symptom. We have to control it on different area. So the technology is evolving still to optimize and influence the brain function. So neural variability, it seems it's the main feature of the brain organization. So, in some of the previous finding, or still we are working on that, it's like a normative data, average brain. It seems we don't have average brain. And neural variability is very, very important, and we have to embrace it. You cannot find two brains to have the same cortical folding. It's all different. And we cannot find two individuals to have the same white matter architecture. So that's totally different. So it's, again, this is a function, structural variability. And also, I'm sure you know about that. So it's even when people are doing the same task, they may use different network. This fine tuning is engaging different area, not on the average. They are tuning for the task. That's different. And not only the task, it means that the symptom is also different. They may have the same level of the depression or anxiety with the same skill diagnosis, but the underlying mechanism is quite different. So neural variability is not challenging. It's actually biological foundation for flexibility, resilience, individuality, and also for personalized brain health. But if we consider neural variability, is that the brain is so variable, then what does it mean? So what would be the problem? So if we accept that the brain is so variable, then why we are talking about inhibitory or excitatory protocol? Because his brain is variable, they always have different response, right? We cannot control that. And if the brain is variable, then doesn't matter if we are stimulating the brain at morning, afternoon, if they are doing tasks, if they are relaxed, or if they are drowsy, if they are asleep, doesn't matter because still brain is variable. And why?
Reza, can I just quickly interrupt? The other thing is, the question is, why can't you go in presenter mode? We have a problem with presenter mode. That's why Reza is doing this screen so he can show the whole slide.
Yeah. So for my next part is what we can learn from non-invasive brain stimulation. Right now, we have very advanced brain stimulation technology that we can control several parameters like intensity, frequency, duration, phase, cross-frequency coupling, targeting, which is like a network stimulation, and we can create different batch of the protocol. So it's this device or the photobiomodulation right now provided us a lot of parameters to control, but at the same time, it caused a problem which parameter we have to use for brain stimulation. The previous study has been shown photobiomodulation it really affects the brain. And how it affects the brain, it has been studied with the EEG. With one of the studies, we show if we are stimulating at 40 hertz, comparing with the sham or active, we kind of even the sham show us the results, but the active was more than sham, and we can induce activity in alpha, beta, and gamma with 40 hertz. Another study, they repeat the experiment, that they find that yes, if you are using the photobiomodulation also with different wavelengths, you can change the cortical oscillation, and it's quite significant in the different frequency band. Another study, again, with the EEG, I'm only focusing on the EEG part, recently by Dr. Jin Chen. They compare different, same frequency, different wavelength, and they find it, oh, wavelength is also important, not only the frequency and the location. And said if you have 1064 nanometer stimulated at 40 hertz, you will have beta and gamma. That's their finding. But if you have 808 nanometers with the same frequency, with the same location, basically you have alpha power increase, not gamma. So it's like it depends also to wavelength. And to answer all of this question, we come with one study here at the Vielight. We are collecting a lot of data. And for each parameter of stimulation, we ask subjects to come 3 times because we wanted to see the reliability of the experiment. Brain is variable, but we wanted to see how variable and based off the variability, how the response is changing. This is one of the experiments we are running for one subject. These subjects, sample subjects, they are coming to receive 20 minutes photobiomodulation 10 hertz. At visit 1, 10 hertz stimulation, it doesn't create really alpha. As you see, this is like during a stimulation and post. Visit 2, also I didn't have alpha. Visit 3, it's quite random. I had alpha, but you see the response to the alpha is quite random. Sometimes we have the more gamma response, sometimes we have more theta response. And lucky in one, I got alpha stimulation, I got alpha modulation as well. For 40 hertz, another subject, as a baseline, they came to receive 40 hertz. I got alpha response. I didn't get anything for gamma. And next day, again, the same. And again, in one of the visits, they responded with the gamma. And I asked the same subjects to come for the algorithm we developed. We call it adaptive photobiomodulation protocol. They are coming at 3 days, visit 1, visit 2, and visit 3. And we stimulated with the enhanced alpha protocol for 20 minutes the same day. And as you see, visit 1, I had alpha. Visit 2, I have alpha. Visit 3, I have alpha. This protocol helps me to exactly induce the power spectrum or the modulation I wanted. The other subjects come for the gamma. That's their baseline, 3 different visits. Visit 1, I have gamma. Visit 2, I have gamma. Visit 3, I have gamma. Also, I have the stimulation of other frequency band. But with the adaptive, with this advanced algorithm, I could induce what I expected. So the protocol we are running is like step 1 of the procedure. We collect the data, we train the algorithm. Right now, with very simple algorithm, we predict the response. We predict the response of the brain base of the different data and feature extraction, the EEG features. We predict the response and we change the protocol and we get what we are expected to get by different frequencies. So it's like a 10 hertz. Alpha is not giving you the 10 hertz, or gamma is not giving you gamma stimulation. Gamma stimulation doesn't give you the gamma response, so it's changing. We are working on step 1 of the project, so it's offline analysis. And then on the second step, we will try to make it closed loop. We are reading the EEG, and through the algorithm, we will change the parameters and we stimulate it and we push the brain during this stimulation, 20 minutes stimulation, to go to the direction we wanted. And in the last part of the project, we wanted to push all of this analysis to the cloud, that people can access to this platform and algorithm all around the world. Like you can push the EEG, and we can control the Vielight device, NeuroPro2, through the algorithm and control it. And I'm going to stop it here, and I will hand over to Marzi.
All right. Thank you. Hi, everybody. Thank you for your time. And, I really appreciate your presence here. And also, I want to personally thank the staff at ISNR for making this happen. So I'm going to present to you the vision that Reza and Dr. Lim mentioned. So, basically, since last year, we have been working on this platform to really give life to the aspiration that we have for personalization. And the platform, it's, I would say, we did an alpha launch, and at the moment it's with the US team for polishing the, interface and user experience. And what I'm going to present to you is basically the general ideas that are driving the design of the platform. And, I hope that you get benefit from this session. So, as, was mentioned by Reza, the personalization is the most important issue that we are trying to address. Our platform does a very sophisticated EEG analysis, getting the raw EEG data, the pre-processing and basic spectral analysis, advanced, analysis on EEG data. But the most important thing that we would like to get, or where we would like to get, is basically the personalized, protocols that will be used in order to, even get to the closed loop part. So this platform is, when we're talking about a Vielight AI platform, the idea is personalization and reaching to that goal. But, there are basically 4 main ideas in the, brain AI research platforms that exist. But I want to mention each and then, explain to you what we are using in Vielight AI platform. So, of course, decoding the signal, there are different ways in which you could use for decoding the signal from pre-processing to processing. And that's one of the passes that we have in our platform. Also, the fact that Reza mentioned, because we know that we have a very diverse users that are using our PBM devices, and we really want to be able to analyze that data and be able to help everybody that is using our devices. So we came up with this idea that we need to have a platform that harmonizes the resources and is able to analyze the data that is coming from different devices as long as they follow the beats format. So we have that feature in our platform, and we understand that, of course, not every data is comparable. So EEG analysis and report is tailored to that specific EEG data that is entered in the platform. Another issue that also Reza mentioned, that is the normative modeling, because this is something that I guess it's missing in the space. And in order to get to that personalization, we really need to be able to have the right analogy of subjects on the platform. So we're using normative modeling to achieve that goal. And we have built the normative modeling, and we are improving our database for the normative modeling. And of course, finally, we would like to close the loop. We would like to be able to do the intervention and also control the brain's response in real time. So just to show you the steps on what we're working on, as I said, we have the measurement of the EEG data. Our platform generates a very sophisticated PDF report. We do the normative modeling. We are working on the personalization, as Reza mentioned. We have the algorithm and the ways that it's possible. Just need to capture the data on our platform and then personalize it for our users, which basically means scaling the algorithm that Reza was mentioning. So we're working on basically control loops and how we can make this loop, close this loop and bringing all these together. So I'm going to show you parts of the platform. So as I said, people can upload their EEG data. Depends on if a user is a single user and they are following specific goals, and toward their goals they can track their goals on the platform. But essentially, like when they arrive on the platform after they submit their report, so people get a dashboard like this. There's an overview. They can see the trends. So if they upload data before and after their intervention, they could see the difference between the two sessions. And also if they upload data longitudinally, basically in different times, they can see the changes across different times. And if they're following specific goals, they can see the plots. They could see the power spectral for their data that they have uploaded. Band powers are also provided, frequency banding details, 1 hertz, we have them all. We also have the brain network. And then also they can download the report. And when they hit download, they can see the report, and that report could also be customized. They could even choose what the part of the report they would like to see. And yeah, so I can show you. Let me see if I can show you the report. So the report is a comprehensive report. That this one, for example, is 29 pages. We have tried to make it somewhat also educational for people that are not too familiar with the concept. So there are explanations of the information that they're seeing on the platform. So it's basically summarized so that for people who would like to show it to their psychiatrist or basically a clinician that is using the platform. So also there's another piece that exists on the dashboard that is an assistant that explains the report for people who would like to know more. They can ask in plain language. It's grounded in the same session, so they can ask questions from that report. And one point to mention that it's not interpreting any results, it's just explaining the results, and it's assistive. We are not positioning ourselves as basically clinical advice at the moment, and we are like a wellness platform. So basically, if the assistant in the backend, it's a multi-agent system that composed of multiple agents that are report interpreter, educator, research specialist, and a scope guard. And so basically orchestrates the output of all these agents and provide the answer to the user. I'm trying to give you a picture of the whole platform. We have a multi-agent system that runs basically the whole platform, and one is basically an agent that does the research for the clinician, one that it helps with the protocol design, and one is that helping with the report, and there's one that knows all the information from the patients that exist on the platform and could really like provide insight on the populations and statistical analysis. But this part will be the next step in our platform that will be launched. If I want to give you a bigger picture of how the platform is built from the back end to front end, we have the data management piece for a single user and our clinicians. They could save the data, they could go back to the data. The reports are safe for them to see the analysis across different times. So that's why we have the database connected. And there's also, analysis service that, as I was mentioning, that generates the report. There's the chat service and multi-agent systems on top of this. So to put it all in a perspective, as I said, we have the platform that analyzes the data, provides a very sophisticated report. As was mentioned, we are going to implement the personalization in the platform, which is the part that we are building. And then also the destination is where we want to go, is the closing the loop, which basically means that we would like to really be able to help users to do the interventions and then also be able to control the amount of parameters that was mentioned by Reza and Dr. Ding. Vielight has more than 30 publications that shows the efficacy of the Vielight devices, but we are also adding normative modeling on top. We also have the cross-device harmonization, and we are working toward the AI personalization that ties to the closed loop together. So as I mentioned, because we are getting ready for the beta testing soon, if you are interested to be a part of the beta testing and test it for yourself, you can scan the screen, the QR code, and be a part of our beta testing.
Well, thank you very much, Marzia. I'll be very brief. The main question is, okay, when can we have it? We are into beta testing now. We want to make sure everything works well. This is the opportunity for you to be part of this journey with us to perfect the platform, and we hope to be able to launch in a few months. But I want to emphasize that this is just the beginning. There is so much that we're doing behind this, particularly in the computational part of it, the algorithms, that you'll see a lot more in the future. So this is not the end. It is beginning. It's very exciting. Okay, Maya, are you compiling the most unanswered, most pertinent questions?
Yeah, Lou, if you open the Q&A box.
Okay, we'll quickly see. Okay, first question is the claim that PBM is robust to within person state variability because it acts on metabolic disposition rather than state? If so, how do you square that with in-between person variability, skull, hair, melanin? It seems like it should hurt light penetration more than it hurts E-field. Okay, we understand that there is research going on independently in still looking at penetration of skull, hair. Literally, there are research groups that have all got data. They are in a process of publication, and I know some are being written up. Some have a lot of data, so they actually answer the variability in the skull. Actually, melanin skin color is really important, and it is actually a factor. Now, having said that, watch out for that. They all mean something. That's why we believe using AI and machine learning is important to nail this down with all the data we have. But I'll say that the one application that kind of bypass a lot of this problem is actually the intranasal. We have data on the intranasal. You just think about it, it's got no skin to worry about, melanin or different barriers. So that is at least about 20 times more efficient than a transcranial delivery of photobiomodulation. Hope that answers it. Have you tried stimulation using a pure sine wave as opposed to square wave, which has harmonic, which would also stimulate the brain? Okay, what we observe, so far, it looks like it is what the brain is responding to. It looks like square wave, and it's kind of harmonized with the pulse on and off all the time. But yeah, I'm not sure what kind of benefit in terms of significance or significant difference it's going to make. But it is something we can investigate into. Is there an open data set for EEG that you are working on in order to develop cluster EEG types for classification and normalization? The answer is yes. Reza, you want to answer that? We're deep into this. Do you see the question? Is there an open data set for EEG that you are working with in order to develop clustered EEG types, classification and normalization?
Yeah, so it's because the data we are training the model, it needs to be with the photobiomodulation. So not many database exists with that. So we collect the data ourselves. We have the Health Canada approval running on the healthy participants, and we're collecting data. But for the REST EEG, yes, comparing with the REST EEG normative data, we always, fan of the neurofeedback, and we run on the parallel quality of the work with the database. But for training the model, we collect our data. So we don't have a lot of data, but every week, we started from last year, we're collecting the data, we train the model on our database, so it's not publicly available.
Yeah, I want to emphasize that we are doing this actually independently because be frank, we don't understand a lot of the, say, normative database out there because we don't have access to they are produced. But we want to collect our own raw data. So you see more and more of that coming. We hope to build it to be a good, strong, credible, and transparent database for our people to use. Am I right in summarizing that, Lisa? Okay, the other question is, Mazia, yes, go ahead.
I saw a question. Somebody is asking if people can track their goals with questionnaires or without any EEG device. I wanted to say yes, that's possible. We are making it accessible to people with no EEG device as well, and they could basically track their progress through answering questionnaires and seeing their progress.
Okay, question
Yeah.
about computer engineering question, interested in computational neuroscience. Personally, I think it's a growing field, especially you go into developing models and now with all the help with AI, I think that's a good future. What is FO?
Yeah, this is a FOOF is a software or it's for frequency for aperiodic analysis.
Is the headband the same as a 19-channel cap? Well, it's actually a different
so
thing. Okay.
for EEG, usually the platform would be, we will accept any EEG, so for giving you the analysis, but to predict the brain response and recommend the protocol for different symptom or condition, we need at least 19 channels data.
Okay, will we have its own brand of EEG headband? Does Mandy also fall into EEG category that could work with the Vielight in this? Reza, you can answer that.
For the moment, we don't have it, but we are working on EEG, so it would be compatible and easy to recommend it. Right now, we don't have any recommendation for any devices.
Yeah, I think as long as we get good quality EEG signals, we can analyze that.
And also, we should mention that people, when they are choosing their EEG devices, they need to make sure that they have access to their raw EEG data. So that's something that they need to make sure they have in place.
We are now doing the back end, but we have there's a study going on at the University of Toronto where we have data based on matching at the same time. We do that ourselves too. So here's the interesting question, because the body and the brain, the body itself generally takes time to respond physiologically. So can we do that? And, we can see the changes by manipulating the PBM and see presented in real time EEG. Yes, but does that mean much in physiological presentation? I think we're out of time. We will find a way to answer these in response after this webinar.
Well, thank you all for your presentation today. It seems like you guys have a lot of things on the horizons and there's a lot of potential with what you're doing. Is there go to the website and ask questions, or will you be attending to those questions separately, I'm assuming? Just to all the participants, thank you guys for joining us. You will get a recording, and I know that we will have a booth at the upcoming ISNR conference. So if you are attending that, please stop by, ask questions, spend some time with our presenters, and they can hopefully navigate you through all of their new materials.
Yeah, we actually have a workshop at ISNR conference, and hopefully we'll be much more developed.
And I do believe that's a little bit longer time frame, if I remember correctly. Well, again, thank you for joining us today. Thank you for sponsoring the registrants today. That was very kind of you guys, and we appreciate your time and your knowledge.
Thank you, everybody.
Thank you.